Thank you so much. I'd love to invite the last two speakers and you to stay on stage, and we'll do a couple of questions and answers. How does that sound? Thank you. You're welcome to sit or stand, whatever is most comfortable. So this should give you a sense. There's quite a lot of variety of projects going on in this field. I think the first question I'd like to kind of get the ball rolling with is for Dr. Natalie Gukasyan. I noticed that you selected the MET, or motivational enhancement therapy, as a sort of therapeutic approach in your research design, and it's a wonderful approach. I'm fully on board with it. But I'm curious if there were other possible therapeutic frameworks that you considered, and if you might have some room for some comparison research, different therapeutic models, and what your thoughts about that are.
So I think the big candidates were, one, just kind of an unstructured approach similar to what some of the earlier studies at Hopkins did, just sort of life review and forming a strong relationship between the participant and the therapist. Toward the more structured end of things, there was ACT, and CBT is actually very commonly used in this population, so that was another consideration. But many of the available CBT models are, like 12 weeks long, and actually we found this very nice brief intervention with MET that actually fit really, really well with the kind of structure we were thinking about.
Fantastic. Yeah. A lot of potential possibilities, but MET is a great one for this kind of research design. I'll ask one question for Dr. Ermakova. I noticed, you have a background too in conservation and in conservation of sacred plants, and I, presume maybe concerned about environment and degradation of environment and the very handsome toads that you've shown us. But I wonder what your thoughts are about the use of in the research use of synthetic 5-MeO DMT for research, and potential implications for the threat to the animals and to their environment that simply may come up as a result of the notice that this is getting in publications now.
I wanted to say that in clinical trials, can you hear me? Yeah. In clinical trials, it's generally easier to use synthetic 5-MeO DMT for various reasons, but the most obvious one is that in the toad secretions, the content is variable. There are lots of other substances, so it's very hard to do clinical research when there's so many compounds in something. And of course, for the toads themselves, the use of synthetic 5-MeO is a lot better in use, because that means that they're not being collected and harassed. And even though people say that collecting toad secretions from the toad could be a sustainable thing, because you don't harm the toad, you catch the toad, express the venom, and then release them. But in practice, it's a very stressful event, and particularly in Mexico, this is done in very big quantities. So people drive with trucks along the road, collect the toads, put them collect them all together, drive them somewhere where they express the toxins, and then release the toads completely random place, not where they were collected. And it also can spread pathogens across different toad populations. If you mix the populations, it can interfere with genetic diversity. So generally, it's not a good experience for the toad.
Thank you. I see we have quite a long line for questions, so why don't we move to some of the audience questions? You guys are getting really great at asking them now, so let's see what we have from the audience.
Hi, my name is Mark Kelly. I work with MindCure Health. I'm just wondering for Natalie, given the challenging experiences that someone with AN, or anorexia nervosa, is working through, were you expecting that they would have equivalent bioavailability of psilocybin in the application? And have you looked at any biomarkers or any other metrics that might indicate that humans are actually accepting the medication in this condition in the same way?
So you're a little muffled. I think your question was about bioavailability.
Yeah, and
So we haven't looked at bioavailability, but we probably ought to. You know, the question is like, is there a problem with absorption? Is this related to an issue with the nutritional status, where there are just levels of like tryptophan or other precursors of serotonin are just like all over the place? Is it to do with like receptor availability, or just sort of like something to do with that? So we don't know. We probably should do it, but it's also kind of hard to do, right? Like, so taking blood a bunch of times while somebody's under the influence is not fun. It's probably aversive. It might adversely affect the kind of experience that somebody has. And it wasn't something we were prepared to do in this particular study, but maybe in a future one, because I do think it's really important.
Thanks.
Hello, my name is Jerry White. I'm a professor at University of Virginia in Charlottesville and an advisor to MindCure in Vancouver. My question is for Joel from UCSD. I myself am an amputee, and for over 30 years, have experienced variations of phantom pain, chronic in the beginning and then sporadic and then fading over time. And it strikes me that it's, or some say it's like grief. If you lose a loved one, it's like losing a limb. And in that first year of pain, it stabs you, it's excruciating, your heart is breaking, much like a phantom limb sensation. Feels like someone puts an ice pick up your foot, and then it may disappear. So most amputees do have experience with phantom pain, but like grief, over time it subsides, integrates into life, and then just becomes sort of a seasonal part of living with limb loss. So I wonder if you had any comment on that, of how you're defining chronic pain, and does it rhyme with grief?
Mm-hmm. Yeah, so we define chronic pain as any pain lasting for more than 3 months, but to your point, in a lot of the different rehab patient populations, whether it's someone who's suffered an amputation or a spinal cord injury or a traumatic brain injury, these patients are grieving the loss of their previous lifestyle, and that 100% plays a part. And my job, myself, what I look for or what I'm looking to do for these patients is to help guide them through that rehab process. And that's kind of the approach I take for patients with chronic pain too, regardless of what it is. I'm walking along a path with them to improve their lives.
Thank you. Let's take another one.
Hi, I'm Daniel, and I'm a practicing psychotherapist. This question is for Dr. Castellanos. So I've admittedly experienced chronic pain, and I've had a lot of improvement with intermittent psilocybin use, but my sense has been that it's actually helped reset my quality of sleep, and that's what's helped. So I'm curious if you had anything to remark on just in terms of discerning between downstream effects versus like primary effects, also knowing that, for many chronic pain patients, anxiety and depression can really exacerbate it. So is there any sense of just that overall picture? Is that something that's being studied?
Yeah, I think it's to be determined. I think that every patient who I interact with who is suffering from chronic pain, it's like an individual, that's why we need to have an individualized approach. My colleague, Fidel Zidane, kind of called patients with chronic pain as having this snowball comorbidity of shit. It's not just the chronic pain. It's the learned helplessness. It's the depression. It's the anxiety. It's, the self-isolation. So it's everything.
Thanks.
Hi, I'm Andrew. I'm a journalist. This question's for Dr. Ermakova. Could you explain what you observed with the people who worsened from 5-MeO-DMT, and do you have any idea of why they worsened?
Sorry, could you repeat the question, please?
For the people who worsened, you mentioned briefly that some people worsened in their symptoms with 5-MeO-DMT. Could you explain a little bit further on that?
I don't really know because those are results from the surveys of people who've taken 5-MeO-DMT in outside the clinical settings. So they've done it in various either recreational or spiritual settings, and it's very hard to know what sort of experiences they had. So maybe they had an experience with not very ethical facilitator of the ceremonies, or maybe they've taken it by themselves at home and had a challenging experience that they didn't have the ability to integrate or figure out. So there could be various reasons.
Great. Let's continue with another one.
Hi, my name is Michael Heiken. Thank you all to the speakers. My question is for Anya. What do you make of the conflicting pharmacokinetic information on 5-MeO based on Jonathan Ott and Sasha Shulgin's work?
In what way is it conflicting?
In that Shulgin thought that 5-MeO was bioavailable sublingually, and Jonathan, or I have it backwards. One of them found it bioavailable, and the other one didn't.
So whether it's orally active or not. Yeah, there is a variety of opinions whether it's orally active or not. So some people say that it's not orally active because it undergoes really first-pass metabolism, and it's broken down by monoamine oxidase very fast. But then there are some people who report that it is orally active. The thing is, we don't really know. Studies in animals show that it's orally active, at least in sheep, but you require a lot higher doses for that.
Great.
Oh, okay. Hi, my name is Alex Birnbaum. I'm a writer, and currently I'm working with a nonprofit in Canada who deals with patients with something called PSSD, which is post-SSRI sexual dysfunction. And Dr. I'm so sorry if I'm mispronouncing this, Gukasyan.
That's good enough.
Sorry. You mentioned something with your anorexia nervosa patients where they were not responding to psilocybin doses. And I'm wondering how you explore something like that because a good chunk of these people they've tried to reverse their condition, which often includes emotional anhedonia, and there's a lot of suicide. But they don't necessarily respond to the medication even in higher doses. And my question would be: A, is anybody willing to look into this as a whole? But also, how do you actually study that? You were talking about the 5-HT2A receptor not being responsive, stuff like that.
So what was the first part of that question? I couldn't hear it because of your mask.
If anybody is either presently looking into PSSD, basically SSRI damage and psychedelics, but also just sort of those, neurotransmitters, those receptors, and what's damaged and how to fix it.
Yeah, so I'm not aware of any lab studies, like any imaging studies that are going on. We actually just wrapped up data collection and are writing up a survey that we did about essentially looking at subjective psychedelic effects from psilocybin after SSRIs have been discontinued, like as Chuck Raisin talked about earlier today. We're seeing that people who had recently been on SSRIs have less of a response to psychedelics, both like therapeutically and also maybe just in terms of what they can report subjectively. So there's that kind of study going on. And collecting data for that, we sort of did bump into this PSSD community online, and they had I think we had some people fill those out. But you could do imaging studies, right? You could do like receptor occupancy studies to kind of see, after somebody discontinues SSRIs, do we see any actual difference in that? Like, is it really permanent damage as people are claiming that it is? And it sounds like for many folks, they do see effects maybe like up to years after discontinuing SSRIs that they feel kind of different. It's not the most common, but it does happen, and it can be pretty devastating when it does. So, but that is one way that we might be able to study it.
Thank you. All right, we're going to close up the questions right there. Thank Thank you again to our three presenters, speakers, for the amazing variety.